Oh, hello. Welcome to Lecture 2. Today we're going to actually talk a fair bit about Mendel's P's and hopefully illuminate some ideas and some concepts that are new for you. All right. So the work that Mendel did to study the genetics of various traits in peas, actually he studied 7 different traits, round and wrinkled, peas, yellow and green. We're going to focus on those first two, but he studied 5 other traits which are shown below. Um, the way in which you actually do genetics is to set up crosses. And so we've been talking about when we do the complementation test, you have to get different strains of yeast and cross them together. In his particular case, he was working with strains of peas that were called breeding true. And by breeding true, meaning that if you have a round strain of peas, no matter what, if you continuously self the peas, you would always continuously obtain round peas. Or if you had a strain that had wrinkled peas, you would self them, you would continuously obtain wrinkled peas. Now, the way in which one does the crosses, it's when I said selfing. Now, obviously, that's not something you can do with most animals, right? So you can't just like take a different mouse, for example, and self it. By selfing, what I mean by crossing with itself. You have to be very careful when you do genetics with peas that the pollen granules from a flower are actually pollinating the stigma which allows the pollen granules to grow down into the ovule. You have to be very careful to make sure that the pollen granules from a certain genotype or from a certain plant fertilize the specific stigma. And so if you're not careful, actually what happens is selfing is actually the most common outcome of pea plants. So if you put a single pea plant in a room and don't allow it to be pollinated, it will self itself because the pollen granules from the anther can fall onto the stigma. Now, you can also help that process, and so in this particular case, one can do a selfing by taking a paintbrush and taking the pollen granules off of the anthers and putting them directly on the stigma. But if you want to set up a cross that is not through selfing, you have to be very careful to make sure that no selfing occurs on those flowers. And so, for example, this particular flower here, you want this flower to cross with this flower. Well, you can easily take the pollen from one flower with a brush and move it onto the stigma of another flower, but you have to make sure that the stigma isn't also fertilized by pollen granules from the same anther, so you remove them with scissors. And so you can do selfing very easily, but cross-pollination is a fair bit of work. Now, in the crosses that Mendel performed, which we briefly discussed in the last class, there was a genotype which I said was breeding true. He didn't have the designation big W, or little w at this time, but there was two different strains of peas, one which always gave round peas and one which always gave wrinkled peas. Now, the observation that he made is that in this particular cross, right, there were all-round seeds, and he proposed there were both of the alleles from the parental strains here, big W and here, little w, and he observed a 3-to-1 ratio, and we're going to actually talk about the mechanism of the 3-to-1 ratio of round to wrinkled and where that comes from. And actually, cutting to the chase, it is rooted in meiosis. Okay, and so I'm actually going to talk a little bit not about genetics, but I'm going to talk about another field which is actually called cytology. And cytology is the study of basically cells or genetic material in those cells through the microscope. Basically, it's analyzing cells through microscopy as opposed to genetic analysis, which is the analyzing phenotypes. Now, for a long, long period of time, it was not actually known, and this was only revealed again in the early 1900s. It wasn't really known that chromosomes carried the genetic material. Now, you could look at cells, even early on in the 1800s, people could look at cells and see that there was material inside the cells, or inside the nucleus in particular, which the name for those materials were chromosomes. So chromo meaning colored, and soma or some meaning body. So colored bodies could be seen inside the nucleus, and one could also see that these squiggly colored bodies inside nuclei seem to move into both daughter cells during cell division. So one could see that a nucleus would divide, and the two daughter nuclei would contain these types of condensed bodies. And at the time, now we of course know that genes reside on chromosomes, but at the time there was, a bit of a debate because, some people clearly argued like, okay, well, genes certainly, the material of inheritance resides on chromosomes. As a matter of fact, some people argued that each chromosome represented a single gene, although it's a little bit hard to understand how that would work. But there was also a group of people that argued that the chromosomes didn't actually code for anything, that they were basically structural, so that they were the scaffold or the structure that basically built the nucleus, and that the genetic material resided off of the chromosomes, presumably in the nucleus, although it could have been elsewhere in the cell. So it's an interesting thing to think about how one actually proves that genes reside on chromosomes, and we'll come back to that in a later lecture. But in order to sort of think about how, which we know genes move around on chromosomes, we actually need to start a little bit thinking about the cell cycle. And so this is a little bit of a cartoon because there's something very important that happens during the cell cycle, both in mitosis and meiosis, and that is DNA synthesis, or the S phase. All right. So basically, any haploid organism any diploid organism will have two chromosomes that come in pairs, and before any cell division occurs, right, the S phase has to occur. So the cell cycle basically and goes like this, and so we start off and cells have just divided, and mitosis has just occurred, and right after cell division, there's this period called G1 or gap 1 in which cells have not divided. The DNA has not replicated. And then can take a long period of time for DNA to replicate. And then in G2, there's a sort of period in which cell division is about ready to start, and the cells are getting ready to engage in a round of mitosis or perhaps meiosis. And so there's this important part in the cell cycle in which DNA is synthesized. And so continuing from a cytological perspective, we're going to have to make a very clear distinction between mitosis and meiosis, which you've heard about, but we're going to actually start putting genes on these chromosomes. We're not going to keep the worlds of genetics and cytology separate. We're going to see how genes move around in this process. So during mitosis, obviously, cell division occurs in what you would call the soma, or the body, as opposed to, say, the germline or the tissues that produce germ cells. And so the typical description goes prophase, metaphase, anaphase, telophase. Prophase is the time period where there's chromosome condensation. Metaphase occurs when the sister chromatids are on the plate. And then the reason there are sister chromatids is because DNA replication has occurred prior to these events, right? And so the sister chromatids come from replicated DNA. Then anaphase, in which those sisters split apart, and telophase occurs when there's a new nuclear envelope established. Okay, so what I want to do, and we're going to switch to actually doing some work on the board, is we want to actually put genes on the cell cycle. Okay, and so I'm going to switch over to the board. We're going to basically walk through how the cell cycle works with genotypes in mitosis, and then we're going to do that in meiosis. So I'm going to quit out of PowerPoint. I'm going to switch to my Zoom screen here. I'm going to make full screen. And basically what I want to do is we're going to walk through how mitosis would produce daughter cells when an individual has a big A, little a genotype. All right, so let's actually go back a little bit further, maybe to a point in which you don't want to necessarily think too much about, but you will need to go back to the point at which the individual that has a big A, little a genotype was produced, and that happens at fertilization. So at some point in history, this would have happened with you or any other living diploid eukaryote. A sperm, which would have the big A genotype, may have united with an egg, right, that has the little a genotype. And if we were to look in there, what that would mean, because sperm and eggs are haploid, right? And I'm going to draw a chromosome that's basically a squiggly line like this, and this would have a big A genotype here. And then I'm going to draw a squiggly line. And we're going to keep it simple. We're only going to focus on one pair of chromosomes that have the A gene on it. We're not going to focus on all the other chromosomes. There's many other chromosomes flying around in this process. We're only going to focus on two. So this would have the little a genotype. Okay, so both of these are haploid because they're gametes. All gametes are haploid. And they're going to fuse together and they're going to produce a diploid. And that diploid will have that particular genotype that we're talking about. All right, and so a diploid genotype is generated. I'll erase this. And I'll just basically show big A, little a, and so there's going to be a squiggly line like that, the big A allele on it, right, and a squiggly line like that with the little a allele on it right there, and this is a diploid. Okay, now this is the very first cell. Now, the very first cell has to produce, well, not all organisms, but let's just talk about, say, for example, humans or mammals. I mean, yeast, it would be a little bit different, but let's talk about larger organisms. They'll have to build the body of the organism. All right, well, what produces the body of the organism? Well, all the cells of the organism, except for one particular class, have the same genotype, right? And the way that they produce genotypes is basically the cells undergo a clonal expansion, meaning every single daughter cell will be, for the most part, genetically identical. And that occurs through mitosis, right? Mitosis, right? And so in that process of mitosis, you are going to build the soma. And I keep talking about soma, but I basically mean all the sort of parts of the organism that don't represent the germline, and the germline are the parts that represent the lineage that produces gametes, sperm and eggs. So you're going to build the soma. And so it's worth thinking. Your thumb is your soma, your ears are your soma, your elbow is your soma, your liver is your soma. Your germline is in the ovaries or the testes. That's germline material. So you're going to build the soma, and this is going to go mitosis and replicate and replicate and replicate. Now I want to be clear about one thing. Mitosis does also occur in the germline, right? Because cells need to expand prior to going through meiosis. And so it's not like the germline has no mitosis. The germline has plenty of mitosis. There's germline stem cells that are dividing through mitosis all the time. At some point, those germline stem cells commit to meiosis and produce gametes. So I don't want you to think that mitosis only occurs in the soma and doesn't occur in the germline. Mitosis occurs in both the germline and the soma. But in the somatic tissues is where mitosis occurs to build the entire structure of the organism. Okay, so let's walk through how this works. So we start with this diploid. We're going to actually erase this. And we're going to see how the cell cycle occurs. So that first individual cell, which has this genotype, big A and little a, right? Now, which part of the cell cycle is this? Well, we would call this gap 1, right, or G1, because it's occurring before DNA replication. All right, so the cell's going to divide, and the first thing that has to happen is S phase. S phase, DNA replication. And we're going to talk a lot about DNA replication later, but for now, we're just going to basically show DNA replication as each of those chromatids. This is one chromatid and one chromatid, but those are actually also designated as chromosomes. Each of those chromosomes is going to replicate, and it's going to produce that kind of famous structure. Oftentimes you'll see it like this, actually. Well, that's terrible, but you get the idea, like a sort of a weird X. All right, I'm going to show it as this, and they're actually joined at the centromere like that. All right. So there's one of the chromosomes, and then there's the other one of the chromosomes after DNA replication. Okay, now this is extremely important, and people mess this up all the time. Okay, this has a genetic identity of big A. This has a genetic identity of little a. And what's really going on here is at the DNA sequence level, which we'll talk about extensively, there's something that distinguishes this allele from this allele. Maybe this gene is mutated or deleted or who knows what it is. But there's something about the DNA sequence that's different here from here. Now, when this chromosome here that has the big A genotype undergoes DNA replication, what are the allelic states of the two sister chromatids? Well, the two sister chromatids are genetically identical to each other. There may be some mutation that could occur, but for the most part, the sister chromatids are genetically identical because they're the result of DNA replication. One strand, one duplex of DNA becomes two duplexes of DNA. So what would be the genotype? Both of these will be big A, right? Don't put little a, big A there, right? Because that would be a mistake. That would indicate that something really was going on weird. I mean, things can happen, and we'll get into ways in which that could happen, but after DNA replication, the sisters are identical. All right, so what would be the genotype of this one here? Okay, well, it would be little a, little a. Right. Okay, now we're going to replicate this cell, and the way I'm going to do this is I'm sort of going to, since I don't have a lot of space, I'm going to kind of go back and forth here. So I'm going to erase this, and then I'm going to say, okay, what happens during DNA replication? Excuse me, what happens during cell division? So DNA replication has occurred. Now we're in G2. And now we're actually going to want to split the cells. All right, and the way that in mitosis, and this is key, this is mitosis, works is that all of the centromeres line up in a column, essentially. I mean, that doesn't literally happen when in the cell, but the way to visualize it is that. So you want to, when you're drawing these types of things, you want to draw a cell like that, or actually, let's be more clear, a nucleus, all right? And you're going to draw, I'm going to keep them a little smaller here. I'm going to draw like that. So all of the centromeres are kind of lined up, and the centromere is a part of the chromosome. I haven't defined it yet, but it's the sort of part of the chromosome in which the spindle fiber is attached to before the cells divide, right? And the spindle fibers attach and then enable the cell division to split the two chromatids apart. Okay, so we have these genotypes, but we just sort of stacked them up. So this is going to be big A here, and this is going to be big A here, and this is going to be little a here, and this is going to be little a here. All right. All right, so we've gone into this configuration. This configuration is called metaphase. All right, and so when I say on a question or something, show something on the metaphase plate in mitosis, you would draw something like that. They're all lined up in the middle of the nucleus. All right, so then what happens next? Okay, well now we're about ready to engage in anaphase. Anaphase is when they're going to split apart, and it's the centromeres that split apart, right? So centromeres will split this way and this way, and centromeres will split this way and this way, and that's going to end up with two daughter products, right? It's going to end up, there's going to be a line, basically, that goes that way, and on one side of the line, right, so that's going to be this side here, right, there's going to be, and the centromeres will split right there and right there. You'll end up with one of the big A's right here, and then since we're talking about this half of the nucleus, we'll end up with one of the little a's. All right, now let's focus on this half of the nucleus, right? Well, that is going to go and make another cell, right? Again, we'll have big A and little a. All right, so this is the most important thing. These are genetically identical to each other and genetically identical to that precursor. Okay. The other thing I want to say is, do we call these diploid or haploid? All right. Now it sort of gets confusing because you see all these lines of squiggly. These are still diploid. All right. What you do is you always count the number of centromeres to count the number of chromosomes, right? And so since before we were saying that when we only have one pair of chromosomes, we would say n is equal to 1. All right. And n basically indicates the number of chromosomes in a haploid complement. But here we have a centromere and a centromere, so we would say that there are two chromosomes. Or we would say that we are 2N, right? So these are diploid. And so I just want to make one final clarifying statement. Cells that are diploid that engage in mitosis will produce diploid cells. Cells that are haploid that engage in mitosis will produce haploid cells. It's because the cell, they're genetically identical after mitosis. And cells can be haploid and engage in mitosis, okay? That would just basically reduce this problem in half. Okay, so there's two daughter cells. So that's mitosis. You maintain ploidy, all right, and you maintain the genetic identity of the daughter cells equal to the parental cell. All right, so we're going to have a little problem here now. I'm going to quit out of the little link here. I'm going to go back to sharing my screen. All right, so let's think about this for a second. If a cell with a big A, little a genotype performs mitosis, the outcome will be these are the options. 2 haploid daughter cells, each with the big A, little a genotype. 2 diploid daughter cells, each with the big A, little a genotype. 2 diploid daughter cells, one with big A genotype, and the other one with little a genotype. And then D, 2 haploid daughter cells, one with big A genotype, and the other one with little a genotype. Okay, let's talk about A. 2 haploid daughter cells. Well, no, because we were diploid before, right? Actually, this answer is nonsensical even for 2 reasons, because it says each with the big A, little a genotype. If an organism is haploid, they only have one allele, right? So you couldn't be big A, little a, and haploid in this context if the big A and little a represent alleles of the same gene. So A is incorrect. Okay, B is the right answer. I'm going to skip to C and D, though, to explain why they're wrong. All right, so another option is 2 diploid daughter cells, one with big A, and the other one with little a. Well, yes, 2 diploid daughter cells would be produced, but they wouldn't be different in genotype like that, all right, because the daughter cells should have genetically identical genotypes. D is also wrong, 2 haploid daughter cells, one with a big A genotype and one with a little a genotype, because a diploid wouldn't produce a haploid genotype. So the answer is B. Okay. Now, meiosis is obviously very different because it doesn't build up all of the cells of an organism. All it does is it's that essential last cell division that occurs prior to gamete production, right? And since gametes have to fuse, haploid gametes have to produce to make a diploid, all right? If there wasn't a restoration of haploidy, every diploid gamete that would come together then would be tetraploid. You know, you'd have this continuous increase in the number of ploidies. So you have to go diploid, soma, haploid gametes, fused together, diploid, soma, so on and so forth. So here's the view of cytology. It's similar in the sense that there are sort of like these 2 parts that repeat. All right, so prophase, that's when what I say the homologs pair. Now, the homologous chromosomes are the sort of chromosomes that come in pairs, one of which comes from one parent and one of which comes from the other parent. All right. So during prophase, the homologs have to pair with one another. We still don't really know how they find each other like this. I did some work for a while to figure that out, and I was not successful. We haven't known for 100 years how this works. But anyway, in metaphase 1, the homologs, and I'll show this more clearly on the board in a moment, the homologs line themselves up on the plate, but this is a big difference. Before I said in mitosis they line up with the centromeres all lined up. Here, the centromeres are going to be lined up in pairs side by side. And in anaphase 1, this is not when the centromeres split. This is when the homologs, the pairs of chromosomes, split apart from one another. Okay? Then metaphase 2, now metaphase 2 is similar to the metaphase that I showed of mitosis, in which the sister chromatids are all lined up on the plate, kind of like the centromeres are all lined up. And in anaphase 2, that's when the sister chromatids separate, all right? So they have to obviously still do DNA replication before engaging in meiosis, but the sister chromatids don't separate until the second round of meiosis. And then in telophase, all 4 of the haploid products get produced. Now, it's worth thinking about in which point of this cytological view of things does ploidy go from diploidy to haploidy. And the answer, you could argue, would basically be anaphase 1, because that's when the homologous chromosomes separate and now they're not paired with each other anymore. Okay, so we're going to walk through how a lot of that works. Now, there's a particular part of prophase that has these designations called leptotene, zygotene, pacotene, diplotene, and diakinesis. And those are actually basically cytological terms that sort of describe the degree of compaction of the sister chromatids. Diplotene and diakinesis are basically when crossing over is beginning and starting to resolve. All right, and we'll talk a lot about crossing over shortly. Okay, so now we're actually, instead of marching through cell division of mitosis with a big A, little a genotype, we're going to actually walk through meiosis with a big A, little a genotype and talk about how that differs from mitosis. So we're linking cytology with genetics because we're following chromosomes. And so I'm going to actually skip out of this screen now. Whoops. I have to do escape. All right. And then I'm going to go back to my little zoom. And we're going to walk through meiosis now. With a big A, little a genotype. I just want to be clear about something. We've moved from Mendel to all this stuff. All of this stuff is what's going to explain Mendel. And we could go through all the Mendel without doing the cytology, but it seems to me it always makes more sense to sort of think about Mendel and the cytology and the chromosomes all at the same time. Okay, so we're going to start at the same position, right? We're going to start off with this sperm cell that's haploid, and we show it like this, a little squiggle with a big A on there, and then there's an egg cell that's also haploid but has this particular genotype, little a. All right, and so we're going to generate that diploid cell now, all right? And it's going to be like this, big A and little a, right? And now we are actually going to start. What would happen prior to meiosis in the germline? Well, typically, actually, this would be a germline stem cell that would actually make many, many copies. So originally, prior to meiosis, that cell lineage in the ovaries or in the testes, for example, will engage in many rounds of mitosis. Well, let's say we're done with mitosis and we're ready to engage in meiosis. Well, what's the first thing that has to happen? You're in G1 now, all right? And so I'll go back to this over here, right? Your big A and little a, all right? And what has to happen? Well, S phase, right? S. And DNA replication has to happen, and you're going to basically generate the same configuration that we did before. You're going to basically show these chromosomes as kind of like these sort of squiggly X's, right? And the sister chromatids, these are sister chromatids and these are sister chromatids. What are their genotypes? Okay, well, this is big A. What is that genotype? Yes, it's big A, right? If you put little a there, that would be a mistake. Why would that be a mistake? Because these two sisters are the descendant of that, and DNA replication will produce two genetically identical sister chromatids from one original precursor chromatid. All right, so what's the genotype here? Little a, little a. All right, so now we're going to divide, all right? And this is the biggest difference between meiosis and mitosis, is in meiosis, these are not going to separate the sister chromatids at this point, and they're not going to split the centromeres. As a matter of fact, the way that I've shown it is kind of the way you can imagine it, is actually these are lined up. And so the way that the division is going to occur here is actually, these chromosomes, by the way, big A and little a, have been hanging out with each other for a long time, right? I mean, we're talking in humans, they're about ready to engage in meiosis. they've been hanging out in a mitotically dividing lineage for, who knows, 15, 30, 35, 40 years. I mean, it depends on how old the person is that's producing these gametes. But they've been hanging out with each other for a long time. But now, the ones that came together in that sperm and egg, they've been hanging out for years and years and years. They're about ready to say, see you later. It's been good. It's been real hanging out with you. And the way that the division works, and I'm going to actually kind of use this one, is that this centromere will go that way, and both will go this way, and this will go this way. So now the actual sister chromatids are retaining together. And so what you end up here is you haven't broken apart the sister chromatids yet, right? And over here, you haven't broken apart the sister chromatids yet. So little a over here, big A over here. Okay. Now, that is not the completion of meiosis. Now, it's a fair question to say, are these haploid or diploid? You might say, well, there's two sister chromatids, so that they're diploid. No, they're haploid here, because you count the chromosomes by the number of centromeres here. And so there's really just, I mean, technically there's two sister chromatids, but technically there's just one chromosome there. There's one of those progenitor chromosomes. So these are haploid at this point. Even though they have two sister chromatids at this stage now, here and here, they're haploid. Okay, so what happens next? Well, what happens next is basically like the mitotic division. And now the sister chromatids are going to separate from each other. And so this is, you call this M2, metaphase 2. The way we have this lined up, M2, metaphase 2 is the second metaphase. The first one I showed was metaphase 1. And now the sisters will separate from each other, right? And so you'll generate chromosomes where these things rip apart, and you end up with big A, right? This is where they break apart. One goes this way, one goes that way, but I'm showing them both of them here. And then these sisters here will break apart, and you end up with little a and little a. Okay. And so how many products do we end up with? We end up with 1, 2, 3, 4, and they're all haploid. Now remember, the outcome from mitosis, I'm just going to write this up here, mitosis, I'm going to put it in a box so it's not unclear, didn't look like that. What the outcome of mitosis looked like was you retained those two chromosomes because you retained diploidy, but you had big A, little a in there. Okay, so we're going to walk through some additional kind of questions. So I want you to think about this. If a cell with a big A, little a genotype performs meiosis, the outcome will be, and then it's either two haploid daughter cells, one with big A genotype, other one with little a. Well, that's wrong, right? Because you should have 4 haploid daughter cells. 2 diploid daughter cells, one with big A, and the other one little a, little a. Well, that's wrong because you're not going to end up with diploid daughter cells. 4 diploid daughter cells, 2 with a big A genotype and 2 with a little a genotype. I mean, that's wrong in 2 ways. First, you're not going to end up with diploid daughter cells. And then the other one is, those aren't even diploid daughter cells as stated. Those are actually haploid. So D is the right answer because you end up with 4 haploid daughter cells, 2 with a big A, right, which I showed in the previous work on the board, and 2 with the little a genotype. Okay, now there's some interesting things to think about because actually we always are saying like, oh, there's the 3-to-1, and we're going to show you exactly how the 3-to-1 comes from meiosis. But it's actually kind of an interesting thing to think about. So did he actually observe a 3-to-1 ratio? All right. Well, in fact, he observed the ratio of 5,474 to 1,850. And that's not exactly 3-to-1. Okay, that is, in fact, about a 2.96 to 1 ratio. Now, you might be saying to yourself, well, you're splitting hairs. I mean, that's ridiculous. It's clearly 3-to-1. And what you're really doing when you say that is you're saying to yourself, well, maybe it technically is 2.96 to 1, but it's a very probable or likely outcome to achieve something sort of like close to 3 to 1, but not exactly 3 to 1. In fact, you might argue like, well, the exact chance of a 3 to 1 ratio is going to be small. And if you actually only collected like 10 progeny, if you think about it for a second, you couldn't get a 3 to 1 ratio, right? Because you'd have like 7.5 to 2.5, that it doesn't work. The way we need to think about this is we actually need to think a little bit in terms of probabilities. And you can say to yourself, well, it's probably pretty likely that it's a 3 to 1 ratio, even though it looked like 2.96. And so the way we need to think about that is to think about what is the probability of observing this. And actually, we're going to, we're jumping around a little bit here. We're starting with Mendel, then we're going to cytology. But we're actually going to think quite a bit about now. And this is going to be something we spend a lot of time on for the next 2 or 3 lectures, is thinking about probability. Okay, because actually probability is at the foundation of genetics in a significant way. And so the probabilities that we're going to start talking about now are binomial probabilities. And by binomial probabilities, I'm saying that there's either this or that, right? So it's either heads or tails. So we're going to actually develop a framework to actually calculate probabilities of genetic outcomes. And so these are the types of questions we're going to be able to answer when I give you the framework for thinking about probabilities in this way. Most formally, from the biggest picture level, I would say that what we're talking about here is what is the probability of having X certain number of events in n certain numbers of trials, each with a probability of p. Okay. Now that sounds kind of formal, but just sort of zoom down onto practical applications of this, you can ask the following questions. You could say, okay, if you flip a coin 10 times, what is the probability of 10 heads in a row? And you might be able to sit and sort of think about it for a second. You might be able to come up with the answer. But you can ask other types of questions which are related to binomial probabilities. In a family of 10 children, what is the probability that 3 of those 10 are boys? Okay, well, that might be a little trickier. Maybe you know how you might do it. If you don't, that's fine. I'm going to explain it. In a cross between, so now we're getting kind of towards genetics. In a cross between members of the F1 generation, what is the probability that all 20 seeds collected would be round in Mendel's crosses, right? He might think like, wait, I expected 3 to 1, but I really got 20. So what's going on there? All of them round. What? And then this actually is where it breaks. This is where it connects to human biology. If a mother and a father both carry a mutation for cystic fibrosis, what is the probability that none of their 3 children will get the disease? And this is a type of question that actually is something that people think a lot about if they find out that they're a carrier for cystic fibrosis allele. Okay, so to cover this sort of framework for basic probability, I'm going to do some more work on the board. And we're going to talk about thinking about independent events that are independent of one another, and mutually exclusive events, which are not at all independent. And we're going to talk about how you count binomial events. And then for this lecture, we're not going to get to it, but we're going to talk shortly about conditional probability. Okay, so I'm actually going to switch back to the board for a bit. Okay, now when we are talking about probability in this context, we kind of are going to use a framework where we say something like this. P and then parentheses A. And what we mean by that is basically what's the probability of some event A happening, right? So you could say, if you have a child, what's the probability that that first child is a boy? You could keep it simple. I mean, you could write probability first child is a boy, but that would be a little bit extra work. So you could say, well, that's 0.5, right? I mean, it's not exactly 0.5. There's some sort of differences in fertilization rates because of X and Y sperms are weighed a little bit differently. But So, but for now, let's just stick with 0.5. Now, actually, let's think of an interesting scenario. Let's think of a scenario in which a mother and a father have 9 children. Okay? You have 9 children, and they're all boys. Every single one of them. First one, second one. Blah, 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 blah. Okay. They want a girl. Actually, a study was done fairly recently, and there's an excess of families that have runs of one sex followed by only one of the other sex, and that's because people oftentimes will want to have a gender balance in their families, and so they sort of keep trying for either a boy or a girl, depending on what the other ones are. So there seems to be a funny result there in recent populations of families. But anyway, that being said, if you have 9 boys in a row, and then a family has another child, what's the probability that that 10th child is also a boy? Now, if you talk to some people, they might say, well, they've had so many boys, it's very, very, very unlikely to have another boy, so they're definitely going to have a girl. But that's not how it works, right? And that's because those events are independent of one another. What has happened before has no influence on what happens later. Now, there may be some weird things in certain cases in some human families, where there's some sex ratio, what is called sex ratio distortion. But for the most part, 99.999% of the time, all of those events of boys and girls are independent of one another. It's like flipping heads, right? If you flip 10 coins, if 9 are heads, and then you flip the 10th coin, well, it's still 50-50. And that's because those are independent events. Now, the significance of independent events is you can actually calculate probabilities of complicated events, which are a series of independent events, right? And so, for example, if you wanted to say the probability that you got in a family of 1, right? So here's going to be a family of 1, right? The probability that you get a boy, right, is equal to 0.5, right? But in a family of 2, right, the probability that you have 2 boys, because they are independent events, all you need to do is actually multiply the probabilities of those 2 independent events, right? So it would be, in this particular case, simply the probability of 1 boy times the probability of 1 boy being, for the second one, right? So you can just multiply those, and that's the thing about independent events, because the outcomes do not depend on what happened before, right? So this is simply 0.5 times 0.5, which is 0.25. All right, and that framework now can be used, for example, for if instead of 1 child or 2 children, there are 10 children. All right, what's the probability that all 10 are boys? It's simply equal to 0.5 times 0.5, so 0.5 to the 10th power. Okay, so that's fairly straightforward, and you can calculate those probabilities that way. But this doesn't actually let us sort of answer these other questions, like what about if there's 1 boy or 2 boys or so on and so forth. This is just a very simple case where we're looking for a string of events. All right, so to get to actually those sort of more complicated things, we need to think about what are called mutually exclusive events. All right, so mutually exclusive events. All right, so let's suppose a scenario where there's 10 children, right? All right, I can think of 2 mutually exclusive events in which all of them have just one sex, right? There are 2 of those, right? You could actually have all just a whole run of one sex. There's 2 ways, right? It's the probability that all are boys, right? Or it can be achieved in another way, the probability that all are girls. All right, so what we're asking is what is the probability that 10 children, all of one sex? And you can actually see that the outcome of all being one sex is actually comprised of two ways that that's possible, right? One way that that's possible is that all of them are boys, or another way that that's possible is that all of them are girls, right? So you can come up with composite events, right? Sort of you define a sort of outcome that is broken up of different other possibilities. Now, sometimes those possibilities are mutually exclusive, and if you actually make them mutually exclusive, you can, or sorry, not make them, ensure that they are. The way that you can calculate the probability of composite events, for example, the probability that this is the case, is you can just add up the mutually exclusive events. So, for example, the probability of all of one sex is going to be simply equal to the probability that all are boys plus the probability that all are girls. And that's because if you are all boys, you're definitely not all girls. And if you are all girls, then you're definitely not all boys, right? So those things can't be happening at the same time. So if you break down a composite probability into the subparts that are all mutually exclusive, you can add them up, right? So sometimes people get a little bit confused, like when can you multiply and when can you add? The rule of multiplication is if you're building up a probability of a bunch of different events that happen independently, you multiply. But if an event is a composite of a bunch of things that are not independent, and in fact they're mutually exclusive, because clearly these are not independent, right? If you are all boys, then you exclude all girls. So those are not independent, right? They're highly dependent on each other. Okay, so you can say that that is equal to, this is 0.5 to the 10th, right? And then that would be also 0.5 to the 10th, right? So you could say it's 2 times 0.5 to the 10th if you want. Or you could simplify it more. I think it will break down into just 0.5 to the 9th. Okay. Now, from a sort of large theoretical perspective, there are two very interesting and highly mutually exclusive events in the abstract sense. One event is A, and another event is not A. And that's actually, those are mutually exclusive because if A happens, then not A happening is almost the opposite of that, right? So if it rains tomorrow or it doesn't rain tomorrow, those are things, if it's above 90 degrees tomorrow or it's below 90 degrees tomorrow, those are clearly mutually exclusive, right? And you can add them up, right? So you could say, okay, I'm going to find a composite of events, which is the probability of A, plus the probability, and not A, we often just put a little line there, probability of not A. Okay, now what do you think that adds up to? Well, this is the realm of all possibility, right? A and not A basically cover everything that could possibly happen. And this probability is going to add up to 1, right? The probability that my name is Justin plus the probability that my name is not Justin. That covers all the possible names in the world, okay? And so that would actually add up to 1. Now, that's actually kind of a useful fact for the following reason, all right? You can do a little bit of simple algebra. You could say that the probability of A is equal to, and then we're going to subtract this, 1 minus the probability of not A. And that's a useful trick for this particular problem. What if I asked you about a family of 10 children? I'm going to erase this part. A family of 10 children. And I'm not going to ask what's the probability that all are boys, or the probability that all are girls, or the probability that all of one sex, which you could break down to all boys or all girls. I'm going to say that what's the probability of at least one girl? And you can break that up for a second. You could say, well, that, probability of at least one girl includes there is one girl, or two girls, or three girls, or four girls, or all the way up to 10 girls, right? And so there's a lot of, and those are actually mutually exclusive events, but I don't have the framework for showing you at this exact moment how you would break that down. But what does that not include? Right? That larger probability doesn't include this, right? That is not equal to the probability of all boys, right? So you could kind of think of this as, you could think of this as the probability of A, right? A now is the event probability of at least one girl, and this then would be the probability of not A. Okay, well, I want the probability of at least one girl. I want that. Now that's going to be hard to calculate, maybe, because you have to calculate one or two, but this one is easy, right? The probability of all boys. The probability of all girls, at least one girl, is going to be equal to 1 minus the probability of all boys. All right, well, what was the probability of all boys? Well, we already figured that out. This here is equal to 0.5 to the 10th power. So the probability of at least one girl is 1 minus that. And that's going to be a useful trick that you're going to want to be able to sort of be familiar with and enough to think about. Okay, now we've been talking extensively about, or hinting at, there's sort of other things that matter besides like just all boys or at least one boy or at least one girl, sort of those weird scenarios. There are other scenarios that we might be interested in as not at least one girl. What is the probability in a family of 10, all right, that there is exactly one girl? Okay. So that's an interesting thing to think about. So there's an exactly one girl, and you might be saying like, well, could it just be 0.5 to the 10th? Well, that would be incorrect. And the reason that would be incorrect is because that's kind of implying that there's only one way in which it could happen, right? Well, we're going to imagine these slots. There's 1, 2, 3, 4, 5, 6, 7, 8, 9, 10. There's 10 slots there, right? And this is, all the 10 children, and we're going to designate boy or girl status. And so if there's exactly one girl, well, you can actually imagine one way in which it would be exactly one girl. Let's say that this first one here is a girl. Right? That one's a girl. And then if there's exactly one girl, then this would have to be a boy, and this would have to be a boy, and this would have to be a boy, so on and so forth. Now, you have already the framework for calculating that probability. All right, you already have that. Because the probability that that first one happens to be a girl is a half, right? And you're just using the independent probabilities to calculate the sort of like series of these independent probabilities. It's a half for each one of those, right? Half, half, so on and so forth. So this particular outcome would be 0.5 to the 10th. But it turns out when I say exactly one girl, so this is actually a composite of a bunch of mutually exclusive ways in which there to be exactly one girl. Because there's another way for there to be one girl, right? That other way, I'll put these slots again. Other way would be for this to be a boy, and then this to be a girl, and then this to be a boy, and so on and so forth. And you can see how you could figure this out. You could say, okay, let's imagine the first one's a girl, the second one's a girl. This is going to be 0.5 to the 10th as well. All right. And how many ways? Well, there are actually 10 slots for that one girl to go in, right? So you go dot, 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 all the way down, dot, 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 dot. The last one is a girl. All of the other ones are boys. All right. This is that last way in which you could have there to be exactly one girl. So how would you actually enumerate now the probability of exactly one girl? These are mutually exclusive events, and so it's simply the sum of those things, right? Because you can add up a composite of mutually exclusive events to get the probability of that composite. So it would be, this plus this plus this, or simply 10 times 0.5 to the 10th. Okay, now, that seems fairly straightforward. But it gets a little bit wonkier because I could say, well, that's just the case of 1. I can kind of like brute force it in my head to get there for exactly 1. But what if there were 2? What if I were interested in this? Now I don't want the probability of 1 girl, I want the probability of exactly 2 girls now. How do I do that? All right, that's a little bit tricky, all right, because you could say, well, maybe the first one's a girl, and the second one's a girl, and then the rest are boys. So boy, 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 right? All right, and then again, just because these are all independent events, the probability is still is 0.5 to the 10th for that particular arrangement, right? And then you could say, okay, well, let's think of another arrangement. This one's a girl, and then boy, and then girl here, boy, boy, so on and so forth. And you could kind of like, iterate the position of all of these individuals, right? You could say like, okay, well, the next scenario is it goes girl, boy, boy, girl, and I'm going to take this girl, and this is sort of the second girl. I'm going to iterate her down the line. I'm going to count up all of those possibilities. And then you could say, okay, well, then I've counted all those. And then this one here, I'll make a boy. Then this one will be a girl. Then this one will be a girl. And then those will all be boys. And then this one will be a boy. Okay, you can see where this is going. It's a pain in the butt. All right? So how do you solve this problem? Okay, now clearly if you were, if you spent some time, you could tabulate all the ways in which there's exactly 2 girls. Right? You could do that. And then it would just be simple because it would be all the ways, whatever that is, all the ways for 2 girls multiplied by the probability of any particular arrangement. And we know all the arrangements are going to be the same because they're all of these sort of 50-50 combinations all just being iterated. So all of those arrangements, so it would be 0.5 to the 10th. So clearly the problem lies in this. How the heck do you calculate all the ways for 2 girls? All right, and this is a very famous result in combinatorics, and I'm going to tell you what it is. There are 2 numbers you need to know here, right? There is something that we call n, which is equal to the number of trials. All right, and in this particular case, n is equal to 10. So we've been using 10 so far. And by trials, I mean like how many children, or it could be how many times did you flip a coin, or it could be how many pea plants did you collect. All right, so n is the number of trials, and then x, we say, and this is just sort of historical, we call it the number of successes. Right. So in this particular case, if we're counting girls, we'd say, hey, having a girl is a success. But, we don't prescribe any sort of like moral weight to that. Okay. So there's a nice little fancy equation for this, all right, to count the number of ways. And so we could say 2, for example, if we're interested. All right. The equation for counting up the number of ways for n trials and x successes is this equation, and I'll present this in a PowerPoint as well, is simply n factorial, right? So that's, if it's 10, it's 10 times 9, so on and so forth, divided by x factorial n minus x factorial, right? So we're going to actually do this for two girls. So the number of ways for there to exactly be two girls. And you could do it for 3 or 4 or so on and so forth. Okay. So what is n? Well, n is equal to 10, right? So it's 10 factorial up on top, and it's 2 factorial down below, and 8 factorial. 10 minus 2, 8 factorial. Okay, now that looks like a bit of a bear. Now I just want to remind you, a factorial is that number multiplied by one lower times one lower, so on and so forth. So this would be equal to 10 times 9 times 8 times 7 times 6 times 5 times 4 times 3 times 2 times 1, all over 2 times 1, right? 2 factorial, right there, times, and then it would be 8 times 7 times 6 times 5 times 4 times 3 times 2 times 1. Now that's a bit of a bear, but you can see that these things are going to reduce in a nice convenient way for you, because all of this 8 factorial disappears here. So 8 factorial is just disappearing. 8, 7, 6, 5, 4, 3, 2, 1 on there, and then same way as here. All right, so what this ends up being is equal to 10 times 9 divided by 2. So 90 divided by 2, or 45. Okay, what the heck did we just calculate? Okay, we just calculated the number of ways for there to be exactly, that should be more clear, 45. The number of ways for there to be exactly 2 girls in a family of 10 children. And so that's the number of ways, but that's not the probability, right? Probability goes from 0 to 1. Probability isn't 45. That's not a probability. So if I ask you a probability, don't just give me like 7. That would be bad, right? So it's the number of ways times the probability of any particular way right? So in this particular case, it was 45, because that's the number of ways, and we calculated it with that n factorial x factorial n minus x factorial. That's in parentheses. And then the probability of any particular way, we already said, 2 girls and then a bunch of boys, or you can calculate it however you want. It's always going to be 0.5 to the 10th. Okay, so this is going to be a tool that you're going to use extensively for calculating some things. So I want to go back a little bit more to some concepts for Mendel before finishing this lecture, and we'll go cover this a little bit more in the review session on Thursday. So where does this 3-to-1 ratio comes from? I just want to remind you, we've walked through meiosis and we've walked through probability, but we still haven't gotten to that 3-to-1 ratio. He found 2.96, but he said, I think it's 3-to-1. And by doing that, what he really did is he actually proposed a model. So this is the model that Mendel actually proposed. 1, he proposed that genes come in pairs, right? So he sort of found out what diploid was, even before any cytology. 2, he figured out, and this is my designation, big W, you could use R if you wanted. Individuals can either be big W, big W, big W, little w, or little w, little w. 3, each gamete, and this is, in his case, pollen or ovules, but it could be sperm or egg, provides only one allele to the offspring. Each gamete provides the allele in proportion to the representation in the parent. So if the parent's big W, all their alleles will be of that type. If the parent's big W, it's 50-50. And then number 5, gametes and alleles are united independently. Oh, independently, huh? Okay, this is actually going to be going back a little bit to thinking again about independent probabilities. And this is actually what is called, this is the basis of the Punnett square. I'm going to skip ahead for a second just to make sure that we're good. Okay. Um, so the basis of the Punnett square, and by Punnett square, you know, you've seen this a million times. Maybe not a million, but you've seen this before. And what this represents is basically a visual calculator for the probabilities of certain events. And by those certain events, what I'm saying is the probability of certain fertilization events. So you probably haven't heard it in that particular way. But if you have an individual that has this genotype, big W, little w, and you have an individual that has this particular genotype, big W, little w, what does meiosis do? Well, meiosis produces haploid gametes, and it will produce a 50-50 mix of the two different genotypes. Now, obviously, if this was big W, it still is going to produce a mixture of gametes, but only of one kind. So when we're talking about these rows here, what we're actually talking about are the genotypes of the gametes. And so this particular genotype, and we could say that this is on the male side, so this would be pollen genotypes. Pollen genotypes. And obviously, since we're talking about fertilization, these would be ovule genotypes, so the eggs. All right, and so meiosis produces two types of haploid gametes. In the case of pollen, two types of pollen gametes. One's with the big W genotype, and the other one's with the little w genotype. But actually, these are kind of like probabilities. So what's the probability that a random pollen granule is of that genotype? Well, that's equal to 1/2. And what's the probability that the other pollen granule, what's the probability of a little w genotype? 1/2, right? And the same thing applies here. So we could produce ovules of big W, ovules of little w genotype, and the probability is 1/2 for that, and the probability is 1/2 for that. Now, what we're showing on this matrix are actually fertilization events. And what we're showing in this upper corner here is the probability that a big W ovule gets fertilized by a big W pollen granule. And those are independent. That's what Mendel said, that what the genotype of one gamete doesn't influence the genotype of the other gamete. So what's the probability that we get a big W and a big W? Well, those are independent events. So it's simply 1/2 times 1/2, which is equal to 1/4. Big W, big W. Okay, well, what's the probability of obtaining little w, little w? The logic holds. We're multiplying probabilities. It's 1/2 times 1/2, which is equal to 1/4. So I know you've seen the Punnett square before, but it's not clear to me always that people, when they look at the Punnett square, they think about it in terms of fertilization probabilities. That's what you're looking at with the Punnett square, and the fertilization probabilities that produce particular genotypes. Now, actually, so this is a case in which the ovule is big W genotype, and the pollen is little w genotype. That's still 1/2 times 1/2, right? Which is equal to 1/4. But that's not the probability of producing a big W, little w genotype. Now, when we show this genotype, we do not care if the big W came from the pollen or the ovule. We are just showing that where we just don't care. So technically, this is in some ways different than little w, big W, right? Because this tells us that this one came from the ovule or this one came from the ovule. But we treat those as if they're the same genotype, which they are. They're heterozygotes, right? So 1/4. And so those are mutually exclusive events. If you're a heterozygote, you could be a heterozygote if mom is big W, or you could be a heterozygote if mom is little w. And so what's the probability that you have this where you don't care? Well, you add up those mutually exclusive events. So this is 1/4 plus 1/4. And so that framework that I've been teaching you about independent events comes into the Punnett square. Now, a couple last points that I want to make. Going back to this question of 2.96 or 3.1, you can collect a bunch of round seeds. All right, so let's actually look at that Punnett square a little bit. I'm going to go back to the Punnett square, actually, excuse me. The ones that are round are these ones, right? Right, and again, that's 1/4 plus 1/4 plus 1/4, so 3/4, right? But it's an interesting thing that happens is that when you get a round P, like that, in that case, you actually, the genotype we would say is W dash, and that dash means we really don't know, right? Because it could be big W, right? Or it could be big W, little w, right? Because big W is dominant in this particular case, and we've talked about that already when we talked about the particulate theory of inheritance. All right, so, we don't know which one of those it is, and so you can actually ask a couple interesting questions. If you were to actually get that number of seeds, 7,324 F2 seeds, and then you get approximately a 3-to-1 ratio, what would be the prediction then if you were to self these ones? Now, those all have the little w genotype. So little w by little w, of course, will always produce wrinkled seeds. But what if you self these ones? All right, because some of those are big W, and some of those are big W, little w. And the prediction then is that actually what fraction of those are going to be homozygous, big W? And the answer there is actually a third, all right, of those top ones, right? Because we've excluded this from the possibility, right? So of this set, 3/4 are going to be round and 1/4 total is going to be wrinkled. But of these ones, right, the big Ws are a third of them, and the big W, little w's, the heterozygotes, are 2/3. And so what you would expect then, and I'll put that up on the Zoom real quick, what I mean by that. If we go back to our Punnett square, all right, so these are going to be the big W, big W, little w, big W, and little w. Right, so we're excluding this as a possibility when we're talking about just the round ones, right? And so each of these has a quarter probability, a quarter that, a quarter that, a quarter that. And so if you added those things up, it's 1/4 to 1/2 for this one, right? So you can see once you exclude those, 1/3 of the remaining will have that particular genotype. And so if you go back, you can see then that if you were to do this particular cross, if these were selfed, all of these would produce wrinkled ones on below, and 1/3 of these would produce only round P's, and 2/3 of that set would produce, again, a 3-to-1 ratio. It's interesting. If you had a pea plant that had round P's, how would you figure it out? Okay, you don't know what that genotype is. It's big W, or big W, little w. How do you figure it out? Well, another thing you could do would be to do a test cross, right? And a test cross is when you cross an unknown genotype to an individual that's homozygous for the recessive allele, and that makes a particular prediction, because if you go back to the Punnett square, you can end up doing something like what I would call a Punnett rectangle. You could actually do a Punnett square for that kind of thing, or a Punnett rectangle. And the reason I call it a Punnett rectangle is one of the individuals is just going to have little w, right? You could show it like this if you wanted to. little w. This is the little w genotype, which is only going to produce gametes of that type, right? And then, so the probability of that particular gamete is equal to 1. And then, so if you're big W, no matter what, you're always going to produce round and round, right? So if you always produce round in a test cross, then you would be big W, big W. But if you had a scenario where this was your cross, big W by little w, you can see in that particular case that half of the progeny, and this again gets that sort of Punnett rectangle, that now it's like little w is up here, big W is up here, little w is here. I just put a rectangle because there's only one possible gamete there. Half of them will be of this genotype, and half of them will be that genotype. So that's the principle of a test cross. Okay, thank you. That was a bit involved. We'll go through a lot of those. We'll do a lot of problem solving in Thursday's class.